HIV, a disease that affected so many in the 1990s. However, its impact still exists today. The Colorado College Biochemistry 2 class will present the biochemical perspectives of HIV. What is HIV? How does it replicate? Why is it so difficult to treat? These are just a few of the questions that will be answered today. HIV stands for Human Immunodeficiency Virus. Unlike human cells that have their genetic material comprised of DNA, HIV is a complex retrovirus that has genetic material comprised of RNA that must be changed to DNA before it can multiply. To enable its replication and infection cycle, it infects helper T cells. These are cells that you'll also be heard called CD4 positive cells. They are a vital part of the human immune system that have been called the generals of the immune system because they call up troops of other cells to go into battle against invading viruses and bacteria. Impairment of helper T cells by HIV leads the body to become progressively more susceptible to opportunistic infections and ultimately the development of acquired immune deficiency syndrome, or better known as AIDS. Patients are considered to have AIDS when they get certain AIDS-defining illnesses or their CD4 positive cell count is less than 200 in a blood test. To go more in-depth about HIV, we will next talk about the different types of HIV and the history of the virus.
HIV variability is the consequence of a very error-prone mechanism of action and rapid viral replication and mutation. HIV-1 mutates around 1 million times faster than the influenza virus and therefore can spread in your body extremely quickly. HIV-1 and HIV-2 were spread to humans through different evolutionary events and therefore affect people differently. HIV-2 is predominantly found in Africa and is much less virulent and therefore leads to less cases of AIDS. HIV-1, however, resides in Western Europe and America with at least 60 million people infected and more than 25 million deaths. The main take-home message is that due to its high mutation replication rate, HIV is a very hard virus to beat because there are so many different replication events that cause different subtypes. HIV was first discovered after acquired immunodeficiency syndrome, or AIDS, outbreak in 1981 after increasing numbers of young, predominantly homosexual men succumbed to unusual opportunistic infections and rare malignancies. The culprit of which was soon determined to be Human Immunodeficiency Virus type 1. Initial cases were found in small cluster groups that showed symptoms of very rare infections due to the weakened immune system. This included small cases of PCP, a rare form of pneumonia, and KS, Kaposi sarcoma, and a very rare form of skin cancer. As described earlier, HIV-1 type M is the predominant effector in Western Europe and the Americas, which means it's the predominant focus of research in clinical studies. However, this fraction only represents about 12% of total HIV infections. So while we are still fighting the battle against HIV, we are fighting a very small front against the battle, and only one of the subgroups is typically examined.
Globally, 36.7 million people are now estimated to be living with HIV. This breaks down to 17.8 million women, 16.7 million men, and 2.1 million children. Among this group, it's estimated that 21% currently do not know their status, and within the U.S., it's 10% that do not know that they're HIV positive. The vast majority of new infections that happen every year come from this group of people that don't know that they are HIV positive. Last year, 1.8 million people were newly infected with HIV, and 1 million people died of HIV-related complications. This figure shows that the rate of infection of HIV has been declining over the last 10 years. Additionally, the number of people dying annually of HIV, shown in red, has also been declining since 2005. Both of these statistics have large standard deviation errors because estimating these numbers has proven to be difficult. In the last year, the rate of infection has decreased by 39%. Both of these trends is largely due to the growing awareness of HIV and HIV infection and the new drugs made available to more and more people every year that we will be discussing later on in this presentation. The majority of people living with HIV are found in Africa and Southeast Asia, which have the highest infection rates in the world. It's estimated that 44% of new infections occur in a particularly vulnerable group of people, including IV drug users, men who have sex with men, people in a prison community, sex workers and their clients, and transgender people. However, this group varies depending on the region. It's worth noting that in South and Eastern Africa, women, particularly young women, are very vulnerable to HIV infections with high infection rates seen in these areas. This graph shows the upward trend of life expectancies increasing for people living with HIV. The dark blue dotted line shows the world trend, and in 2015, the life expectancy for someone living with HIV was 70 years old. There is a marked difference between the world average life expectancy and the populations in sub-Saharan Africa, where drugs are still not readily available. Only 54% of adults and 43% of children are currently taking antiretroviral treatment. In Colorado, there were 11,199 new diagnoses of HIV in 2014. Men are disproportionately affected by HIV in the United States, and 88% of these new diagnoses were of men. In El Paso County, 138 of every 100,000 people are living with HIV, which is similar to the national average.
So, there are a lot of misconceptions about how HIV is transmitted, and this is due to a lack of education and stigmas in our community. So, HIV can only be transmitted through 4 bodily fluids, and these are vaginal secretions, semen, breast milk, and blood. So, just to go through some transmission rates with you all, HIV is transmitted 8 times for every 10,000 exposures in vaginal sex. However, it is 10 times higher with anal sex, in the transmission being 138 for every 10,000 exposures. And so, to talk about blood, transmission of HIV in blood, in sharing needles from drug use or reusing needles in healthcare, there are 63 transmissions for every 10,000 exposures. And with blood transfusion, this rate goes way up. Blood transfusions have the highest rate of HIV transmission, with there being 9,250 infections for every 10,000 exposures. And so, while we were talking about HIV in the blood, HIV can survive as a virus in dried blood for 5 to 6 days, whereas HIV can survive in blood in a syringe for up to 4 weeks. So, whenever you are handling somebody else's blood, always make sure to take the extra precautionary to wear gloves and be careful about that. And so there's a lot of misconceptions also about how HIV isn't transmitted. And so HIV cannot be transmitted through sweat, tears, saliva, feces, urine, or insects and mosquitoes. And that really, a lot of people get this misunderstood and think HIV is airborne or waterborne, but those are all just misconceptions.
There are a variety of methods to detect HIV in a patient. The most common of these is the recognition of the antibody response to HIV infection. Antibodies are small molecules produced by the body's adaptive immune system to counteract infections. Because the adaptive immune system takes time to recognize foreign antigens and develop an antibody response to them, methods detecting antibodies in HIV may take up to 6 to 12 weeks after infection to reliably diagnose the disease. Additionally, CD4 T cells are a subset of immune cells which are targeted by HIV, and so the concentration of these cells can be used to examine disease progression and severity. Finally, there are ways to determine the presence of foreign HIV complexes like its genetic material, RNA, or antigens. As can be seen in this chart, these varying molecules are present in different quantities depending on the date post-infection. Methods studying the HIV RNA in the red line, for instance, are able to detect the infection many days sooner than methods looking for HIV antibodies in the blue line. Most diagnostic methods make use of ELISA. Essentially, a molecule that is known to bind with an HIV antibody will be placed on the surface of a container, after which a patient's sample will be poured in. Assuming the patient is infected with HIV antibodies in their bloodstream will bind to HIV antigens on the surface of this container and cause the solution to change color. In studying HIV antigens, the same method can be used in the opposite way, in that HIV antibodies are placed in the container and the patient's sample will produce HIV antigens, which will bind to these, and the solution will change color. This latter example of testing for the HIV antigens is an example of one of the more rapid testing diagnostic methods, as HIV antigens are present in the body immediately after infection. Once diagnosed with HIV, patients will often undergo a drug resistance test to determine which treatments are effective for that patient's specific phenotype of HIV. Certain mutations among different strains of HIV will be treated by a certain drug with varying degrees of success, and so this stage of diagnosis is important for the optimization of patient care. Additionally, home tests have been developed that are easy for the average person to use. There are multiple home testing options under names such as the Home Access HIV-1 Test System or OraQuick. The home access example involves drawing a blood sample and sending it to an anonymous lab for testing. Alternatively, there are easy-to-use diagnostic tools that simply require swabbing the mouth and waiting approximately 20 minutes. While the OraQuick or other oral diagnostic tools are easier to use, they may take up to 3 months post-infection to reliably detect HIV. Any positive results with the home testing kit should be quickly followed up with a trip to a doctor's office.
When HIV enters the body, it targets the human immune system, specifically the helper T cells that we've been discussing. HIV recognizes these T cells by specific identifying proteins that are found on the outside of the cell. We refer to these as receptors. The first step of HIV entry is the binding of receptors. On the outside of HIV, there are receptors called GP120. 3 of these GP120s come together and bind to a receptor on the human cell called CD4. Once CD4 binds, a change in shape occurs that allows for another protein found on the outside of the human cell to bind. We call this other protein a co-receptor. In order for the next step in HIV entry to occur, it is necessary for both the receptor and the co-receptor to be bound to GP120. This allows a protein found on the outside of the HIV virus, called GP41, to insert itself into the human cell membrane. This causes a fusion of the HIV and human cell membranes, thus allowing for the HIV infectious viral capsid containing the HIV RNA to enter into the human cell.
Before we go any further, it will be useful to have a quick refresher on DNA, RNA, and proteins. DNA is the molecule which carries the genetic code. It contains the instructions that tell your body how to create the protein molecules needed for life. The two-stranded double helix structure makes it stable and resistant to mutations, or changes in the genetic code. When it is time for a cell to make a certain protein, DNA coding for that protein will be copied into RNA by a molecule called RNA polymerase. This process is called transcription. In humans, RNA is a messenger molecule that DNA uses to communicate its instructions with the rest of the cell. It is similar in composition to DNA, but with a single-stranded structure instead of double-stranded. This means that RNA is more prone to mutations. RNA brings the instructions from DNA to a molecule called the ribosome, which creates a protein based on these instructions. This process is called translation. Proteins are the main building blocks in the body. They have complex 3D structures that allow them to act as molecular tools, cutting or gluing together other molecules. Proteins that help carry out chemical reactions are known as enzymes. So, what does all this mean for HIV? The main thing to remember is that HIV is a retrovirus, meaning that the virus carries its genetic code in the form of RNA instead of DNA. RNA is less stable than DNA, which means that HIV can mutate rapidly, with parts of its genetic code constantly changing. This makes it difficult to develop new vaccines or drugs for HIV because its structure is changing so often. After the HIV virion enters a human immune T cell, the virus is uncoated and viral RNA is released. An enzyme carried by HIV, called reverse transcriptase, copies the viral RNA into a double-stranded DNA with the same structure as human DNA. This viral DNA will then enter the nucleus of the host cell. Finally, another HIV enzyme called integrase will insert the viral DNA into the host genome. Now, the viral DNA is part of the human DNA. After HIV infection, the host cell contains the code for making HIV in its own DNA. Human genetic machinery, like RNA polymerase, continues to copy the cellular DNA into RNA like normal. However, the cell now also contains the instructions for producing viral RNA, which will produce new viruses. This genome integration is what makes HIV so difficult to cure. Even if all virus particles were somehow eliminated from the body, host cells would continue producing new viruses. Additionally, this process allows the virus to stay latent in the body, avoiding detection by hiding in the host genome. It is possible to have HIV genes integrated in the human genome without the patient yet showing detectable levels of HIV antigens. This means that a person can be infected with HIV and not know it, increasing the rate at which the virus spreads through the population. This is why it's so important for people to get tested, especially if they are sexually active.
We've just discussed how the HIV genome is inserted into the host genome. After the HIV viral DNA has been incorporated into the host cell DNA, new viral proteins and new viral RNA can be produced. After these viral components are produced, HIV must leave the original host T cell and enter the bloodstream in order to infect new helper T cells. The HIV virus does this through 3 steps: assembly, budding, release, and maturation. In the first step of assembly, an HIV protein known as GAG localizes all elements of the virus to one part of the host cell's membrane. In addition to GAG, important components of these assembling HIV virus from inside the cell include the viral RNA and other viral proteins and enzymes. At this point, all of the viral proteins are large and are connected to one another like beads on a string. They are known as polyproteins. And remember, an enzyme is a protein that helps biochemical reactions proceed. Additionally, we need some extracellular components to be localized to the same part of the host cell membrane. These are the GP120 receptor proteins. As the virus begins to assemble on the membrane, more and more of the GAG polyprotein and other viral components continue to accumulate. This puts pressure and strain on the host cell membrane, and the membrane begins to bulge out, forming what we call a viral bud. Eventually, the bud gets large enough that it forces separation from the host cell membrane. Then the virus is freed and released into the bloodstream. At this stage, the virus is still immature and is not infectious. For the virus to mature, the large viral polyproteins must be cut into small individual proteins. The cutting is done by an enzyme known as protease. Additionally, the viral RNA, other viral enzymes, and the individual viral proteins that have just been cut must be packaged into what is known as an infectious capsid. The infectious capsid is what will be inserted by HIV into a new T cell. Now that the virus is mature, it is infectious and can infect new immune cells within the body.
Now that we understand the structure and the function, much like scientists of the 80s and 90s, we can now explore how prevention and treatment can occur. Now let's analyze how these substrates that are called inhibitors can act upon enzymes to disallow function and slow down the processes of HIV replication. Enzymes can either be inhibited by competitive inhibitors or non-competitive inhibitors. Competitive inhibitors bind to the active site, which is the location where chemistry occurs. Non-competitive inhibitors bind to a location that is not the active site, but still stops the completion of the enzyme by a conformational change in the active site location. The use of inhibitors is used at HIV key steps such as entry, reverse transcriptase, integrase, and protease. First, I'll talk about entry inhibitors. This is the first inhibitor location that could stop the host cell from getting infected completely. This is at the location of the cell surface, and this blocks the membrane fusion of the HIV virion with the host cell. This was discovered in 1994 and can be associated with proteins such as GP120. Next, let me talk about the reverse transcriptase inhibitors. There are two different types, NRTIs, nucleoside reverse transcriptase inhibitors, and NRTIs, non-nucleoside reverse transcriptase inhibitors. NRTIs bind in the active site and are competitive inhibitors. These resemble the DNA of the HIV genome and will not allow for the attachment of the HIV genome to be transcribed from the single-stranded RNA to the double-stranded DNA. NRTIs are non-competitive inhibitors and bind at a location that is not the active site, but these also prevent the attraction of the RNA to the reverse transcriptase. Next is integrase. Integrase inhibitors are one of the newer types of drugs that have come out and have recently come out in 2007. Integrase the protein allows for the integration of the viral genome into the host cell. So prevention of this would block the integration of the HIV virion's genome into the host cell. This is only the first generation of the drug and has come out, and so now the newer drugs will be interesting to understand what future research can be done. Next up, we have protease inhibitors. As remember, protease cleaves the polyprotein that had been previously talked about into smaller proteins to allow for maturation of the HIV virion. This first came about in 1995, and as you can see at the graph shown, black indicates the number of AIDS diagnoses, while the black dashed line indicates the AIDS deaths. And so we can see that in 1995, with the introduction of the first protease inhibitor, that this really caused a lot of development to occur, with deaths dropping significantly. So this was thought of to be as one of the greater achievements with HIV development for therapy, but it's been recently recognized that protease mutations occur at a much higher rate than many other enzymes. So they've been going away from protease inhibitors to other inhibitors. Now, resistance is key to understanding why that the solution for HIV hasn't come about. Mutations occur at great rates in HIV, and this is not only in the protease inhibitors, but in all inhibitors. So it is important to adhere to the schedule of drugs that is administered by a doctor, because not taking drugs or skipping doses can result in mutations, which change the chemistry of the highly specific drugs to disallow for the inhibition of drugs.
As mentioned earlier, HIV is a retrovirus. Therefore, the drugs we use to treat HIV are called antiretrovirals, which Jacob talked about right before me. Standard treatment involves a combination of 3 or more of these drugs and is called highly active antiretroviral therapy, also known as the cocktail. The choice of antiretrovirals differs from HIV-1 and HIV-2. HIV-2 is resistant to NNRTIs and should be avoided in HIV-2 treatment. Current surveillance programs do not report the type of HIV, therefore information and optimal treatment for HIV-2 is limited. In both cases, treatment goals are the same. Treatment aims to slow the progression of the disease, reduce the chance of transmitting HIV to partners, keep the amount of virus in our body undetectable, and prevent secondary infection. Essentially, the drugs want to stop the virus from growing and multiplying. Treatment will usually involve taking several pills a day for the rest of the patient's life. To this day, we still have no cure for HIV. Once a person is infected, they will always have HIV. The virus is able to remain through dormant or latent infection. HIV hides in long-lived cells called memory T cells, which are important for our immune system. If these T cells are infected, they will not be able to be recognized or cleared by our immune system, and the antiretrovirals discussed earlier cannot stop this inactive infection. New latency reversing agents are being developed to force the virus to reveal itself. Using both latency-reversing agents and antiretrovirals, a new approach called shock and kill aims to eliminate the hidden supply of HIV as well as block new infection events. Sometimes HIV medications can cause side effects. The majority are mild and short-term, and a lot of newer medication has a lot fewer and less severe side effects than the older ones. These mild side effects include feeling tired, nausea, and vomiting. Although some side effects can be serious, like kidney failure, liver damage, and heart disease, overall the benefits of taking the medication and being able to live tend to outweigh the side effects for most people, especially as treatment continues to improve. In 2010, the Center for Disease Control and Prevention estimated the average annual cost of HIV treatment in the U.S. to be around $23,000, and the lifetime cost being about $380,000. Costs tend to be higher for sicker patients with low CD4 cell count due to high cost of inpatient care. 65% of this cost will go to antiretroviral medications alone and will account for the greatest portion of HIV treatment. The rest of the cost includes non-antiretrovirals and non-drug services such as routine checkups.
As mentioned previously, there is no current vaccine or cure to HIV. However, there are medications for prevention that include microbicides, which you can apply as a gel to your genitalia before intercourse and have been shown to have a 40% efficiency in not transmitting HIV. If you're in a high-risk scenario such that an example would be your partner is HIV positive, pre-exposure prophylactics are recommended. These are a daily pill that are prescribed and, if used with condoms, have been shown to have a 99% efficiency in not transmitting HIV. In the chance that you do get exposed to HIV, you do have a 72-hour window to take post-exposure prophylactics, which have also been shown to have 95% efficiency in not transmitting the disease. However, this is not always a great solution, as it is sometimes difficult to know exactly when you've been exposed to HIV. Other prevention mechanisms are treating of other STDs, because other STDs can often lead to lesions or sores around your genitalia, which can be exposed to bodily fluids during intercourse. And lastly is the prevention of mother-to-child transmission, and this can be prevented simply through bottle feeding. Another aspect of prevention is the social aspect. In large part, this comes down to sex education. Knowing how these diseases are transmitted and how you can prevent them is a huge step in prevention. For example, knowing how to put a condom on. Another part of prevention is, from the social aspect, is needle exchange programs. These prevent the transmission of needle-to-needle usage between users. And lastly is serosorting. This is the idea that you get regularly tested to know your status of HIV so that you and your partner can make the correct informed decisions when approaching sexual behavior. The last part of prevention is awareness. We are now in the generation that did not see the effects of HIV and the deaths it caused. This can also often lead to malinformed decision making when engaging in sexual behavior. So having a conversation with the severity and consequences of these behaviors is necessary to make a completely informed decision. Also important in awareness is knowing your risk level and the demographic that you're within, and knowing how widely spread it may be within that population. And lastly is exposing stereotypes and fallacies. By exposing these fallacies, it creates a more open dialogue that can ultimately lead for more information to be available.
Hopefully by now, everyone understands what HIV is, how it replicates, and why it is so difficult to treat. The Colorado College Biochemistry 2 class would like to thank the Department of Chemistry and Biochemistry for all their help. Additionally, the class would like to thank the Southern Colorado AIDS Project for all they do in the community for all those living with HIV.